All right. What's up, Hasan? I want to give you the opportunity to introduce yourself.
So I'm Hasan Sulaeman. I went to grad school with Sarah, actually. We're in the same lab for a while. And I studied disease ecology. Recently, I've been working on SARS-CoV-2, just national surveillance, working with the CDC. And I basically work there as a scientific data analyst.
Cool. Is that your first job working in kind of like the viral disease kind of realm?
Absolutely, yeah. When I was in grad school, I worked on chytrids, which are fungus. Moving on to viruses, it's new to me, but it was exciting. It was fun. It was a challenge, you know.
So that reminds me of a recent memory I had with you. I went to your place of work and research, and they strapped me up to a machine, and they were, as far as I knew, they were pulling plasma, and they because I had COVID, and so I had antibodies in my system.
Timeline here is pretty important, and I think when you first came in, it was the first few months of the pandemic, right? Like you were kind of early to get on the bus, and at the time we didn't have vaccines, we didn't have really antiretroviral just supportive therapies, and so the idea of getting plasma from someone who was previously infected was, when you're infected, you get antibodies, and those antibodies, they can actually be put in another person to elicit that protective response. Since then, we know that convalescent plasma, as we call it, is not an effective treatment. So that program's been stopped.
As far as a lot of people understand, which can be a misconception, immune responses are kind of like quantized. It's like one or the other. In talking to you and through kind of our experiences with the disease, we both kind of know that immunity doesn't quite work like that. I was hoping maybe you could expand on kind of like the various pathways to immunity and the various like in between points that you find.
I think it's important to kind of look at the mechanism, right? It's like all proteins are lock and key. And when you think about lock and keys, like even locker, one of those, lockers locks, there's only so many different combinations you could have on them. And with viruses, it's kind of the same thing. When you have these spikes, right, that are used to kind of penetrate the cell, there's only so many combinations that you could have. So there's levels of cross reactivity. So if you've got a cold, it kind of reacts to SARS-CoV-2 proteins as well in like a very slight way. And even SARS-CoV-2 antibody proteins, they have cross reactivity with SARS-CoV-1. But those are all percentages, and it's important to remember that. And that, when there's a infection kind of event, just imagine like you have really like millions of viruses around you. It just depends on your level of reactivity to those viruses that determine kind of whether or not you're going to be immune or whether or not you're going to be infected.
Would it be safe to say, like, if you had a virus that had 25% similarity to like SARS-CoV-2, you would have like not full immune response, but like a lesser, but still an immune response because of that similarity?
It depends on the family of viruses, right? It's for example, coronaviruses are crown viruses. Like they have the spike proteins, and so there has been studies with flu and cold and MERS, SARS. We know that the level of similarity corresponds with kind of the level of cross reactivity. But I think for other viruses, I'm not too familiar with that, so I can't really speak.
That's interesting. And that makes me kind of think about the different kinds of responses you get from a thing like a vaccine. And so part of that conversation about immunity being perceived as this one or the other thing, but it's actually kind of a gradient. How does that kind of relate to responses to vaccines and like the differences of an immunity you would get from like a vaccine versus just an exposure to a virus?
I want to clear up kind of a misconception first, is that I think a lot of people think vaccines are for preventing infection. It's really for preventing severe disease and death from a pathogen. And so with that in mind, vaccines don't usually elicit the same kind of immune response as an infection does. So for example, if you've been previously infected by COVID, you have memory B cell response kind of 6 months down the line, and there's maturation basically of that response, as opposed to if you got like a vaccine dose, you don't really have the same kind of response from your immune system.
So you worked a lot with SARS-CoV-2, and which is a human disease. But I know, like personally, I know that you've had a lot of background in a variety of diseases, not just viruses. I mean, you have your roots in working with like fungal disease that infects like frogs. And so there's this whole branch of kind of the study of viruses and the study of disease that is called disease ecology, right? We came from a disease ecology lab. And I was wondering if you could kind of maybe expand on what disease ecology is and how things like a frog disease can actually give us good information for predicting and looking at diseases in human populations.
I think I want to start with kind of what disease ecology is, right? I think disease ecology is kind of just, if you think about epidemiology or just the study of kind of how diseases transfer kind of in human populations, I think I'd like to think about disease ecology as kind of that, but with extra bells and whistles, where we also care about how that disease is either kept in a reservoir in the environment or in a species of animal, or just how it kind of spreads in natural settings. So it's essentially kind of epidemiology, but taking the environment as kind of a variable in that calculation. That's how I think about it. And it matters to understand disease ecology working on SARS-CoV-2, because I think it gave me insight on just kind of, we're not in vacuum. It's like, as much as we'd like to think that we live in cities and whatnot, I mean, there's natural environments around us everywhere, in every city. And that affects how diseases get transferred. That affects the infection rate. That affects a lot of things, not to mention weather, climate, etc. Those are all things that we kind of try to look into in disease ecology. And so, yeah, I think if I hadn't worked in a disease ecology lab, I think my modeling approaches for SARS-CoV-2 and kind of just trying to understand it better in a spatial and temporal way, that aspect would have been a lot more lacking.
How did you get involved in a disease ecology lab? Was that kind of where you were headed to as an undergraduate, or like how did your kind of relationship with that lab develop?
I was sitting in my PI's, previous PI's, principal investigators class, and I just read up on him, and I thought, oh my gosh, he does awesome research. And I really just came up to him after class one day and I was like, hey, can I volunteer in your lab?
That's as an undergrad.
Yeah. And for grad school, that for me at least, my undergrad work kind of translated immediately to that. Anyone who's looking for either a master's or PhD program, it bears just kind of looking through someone's repertoire of research, talking to them, and really just going for it. That's how I would put it.
You're not just a disease ecologist. You also have other interests in biology. And I was hoping maybe you could talk about some of your kind of like interests that are more organismal, like taxon related.
Yeah. I mean, I've bred reptiles and sold them for since I was, second year of middle school. So I think that's like what, at 15, 14 years old. So yeah, I've been breeding ball python morphs. I've had tegus, just really a lot of other species as well, like retics, reticulated pythons, blood pythons.
Maybe you could just like briefly talk about like fully your complete journey to being in San Francisco, being this disease ecologist, working as a researcher. How did you get here?
My journey was just really like, ever since I was a kid, I loved this kind of science, research, animals. And so as I got a little bit older, I was just like a gutsy kid who didn't have any shame. So I kept just coming up to people that I really looked up to, either whether they're in science or animal husbandry or whatnot, telling them that I want to learn, I want to know more, I want to be involved. High time for college. I was just like, I'm going to do zoology. After that, I think it was just, one thing led to another. I don't really have like a deterministic answer to how we got here.
It always sounds perfect when we like think about it backwards, but then when you actually think about what you were doing at the time, you're like, man, I didn't know what I was doing. I just kind of ended up here now, and that makes
Yeah,
sense.
yeah. I got into COVID research because it was just, there was a pandemic going on. I definitely thought that I had something to offer, to be able to help, right? And so that's kind of how I got into COVID.
Is there anything that you feel like would have been like good to know, or you would give your like advice you would give yourself as an undergraduate, like now?
Don't procrastinate. I think that's kind of universal, but also just don't. The worst they could say is no. It's like asking someone out on a date. It's like the worst they could say is no. Um, it's like if you're interested in a lab or interested in opportunity, go for it. Like really, go for it. The biggest regret that I have is not going for more things. Really just kind of chasing opportunities down. And yeah, I think if I could talk to my former self, I would tell him to be a little bit more upfront. Or just more brave.
The romance of betterment.
Yeah.
Well,
Love that.
thank you for your time and for
Yeah.
talking about disease ecology and your job studying SARS-CoV-2. And thank you for all your work.
Yeah. Thanks.